The skin and body senses include the sense of touch, kinesthesis, which is muscle movement, and equilibrium. The last two are called body senses because they inform us of the body's position and movement. They tell us, for example, if we are running or standing still, lying down or sitting up. The tactile senses. Touch is important to us in many ways. Sensitivity to extreme temperatures and to pain enables us to escape external danger and alerts us to disorders within our body. Tactile sensations are also a source of many of life's pleasures, including sexual orgasm. Massage enhances newborn babies' development. Conversely, it has been shown that a lack of tactile contact with a caretaking adult retards physical, social, and emotional development. Humans are sensitive to at least 4 tactile sensations: pressure, touch, pain, warmth, and cold. These sensations are conveyed by receptors in the skin and in our internal organs. Mixtures of these 4 sensations form the basis for all other common skin sensations, such as itch. Considering the importance of our skin senses, surprisingly little is known about how they work. The skin, a multi-layered elastic structure that covers 1.7 square metres and weighs between 2.7 and 4.5 kilos, is the largest organ in our body. As shown in figure 5.19 on page 167, it contains a variety of receptor structures, but their role in specific sensations is less clear than for the other senses. Many sensations probably depend on specific patterns of activity in the various receptors. We do know that the primary receptors for pain and temperature are the free nerve endings, simple nerve cells beneath the skin's surface that resemble bare tree branches. Basket cell fibres situated at the base of the hair follicles are receptors for touch and light pressure. The brain can locate sensations because skin receptors send their messages to the point in the somatosensory cortex, or somatosensory cortex, that corresponds to the area of the body where the receptor is located. The amount of cortex devoted to each of the area of the body is related to that part's sensitivity. Our fingers, lips, and tongue are well represented, accounting for their extreme sensitivity to stimulation. And there's no somatosensory cortex map or image in this textbook for some reason, so it's good to look it up. It's sort of this weird looking deformed person to show which parts of the body use more muscles and has more sensation than others. Sometimes the brain locates sensations that cannot possibly be present. This occurs in the puzzling phantom limb phenomenon, in which amputees experience vivid sensations coming from the missing limb. Apparently, an irritation of the nerves that used to originate in the limb fools the brain into interpreting the resulting nerve impulses as real sensations. Katz and Melzack studied 68 amputees who insisted that they experienced pain from the amputated limb that was as vivid and real as any pain they had ever felt. The pain was not merely a relocation of what pain used to feel like in the limb, it was actually experienced in the present. The phantom limb phenomenon can be quite maddening. Imagine having an intense itch that you can never scratch or an ache that you cannot rub. When amputees are fitted with prosthetic limbs and begin using them, phantom pain tends to disappear. Pain. Pain receptors are found in all of the body tissues with the exception of the brain, bones, hair, nails, and non-living parts of the teeth. Free nerve endings in the skin and internal organs respond to intense mechanical, thermal, or chemical stimulation and then send nerve impulses into the spinal cord, where sensory tracts carry pain information to the brain. Once in the brain, the sensory information about pain intensity and location is relayed by the thalamus to the somatosensory and frontal areas of the cerebral cortex. Reflecting the adaptive value of pain, brain recordings reveal that cerebral processing of pain occurs faster than for other kinds of tactile stimuli, permitting a more rapid response. Other tracts from the thalamus direct nerve impulses to the limbic system, which is involved in motivation and emotion. These tracts seem to control the emotional component of pain. Thus, pain has both a sensory and an emotional component. Suffering occurs when both painful sensations and a negative emotional response are present. Spinal and brain mechanisms. Gait control theory, which is a key term, developed by the Canadian psychologist Ronald Melzack and physiologist Patrick Wall, was a major advance in the study of pain. Gait control theory proposes that the experience of pain results from the opening and closing of gating mechanisms in the nervous system. Events in the spinal cord can open a system of spinal cord gates and allow the nerve impulses to travel toward the brain. However, other sensory input can partially or completely close the gates and blunt our experience of pain. For example, rubbing a bruise or scratching an itch can produce relief. Gait control theorists also suggest that acupuncture achieves its pain-relieving effects because the acupuncture needles stimulate mostly tactile receptors that close the pain gates. From a psychological perspective, perhaps the most intriguing feature of gait control theory is that nerve impulses in fibres descending from the brain can also influence the spinal gates, thereby increasing or decreasing the flow of pain stimulation to the brain. This central control mechanism allows thoughts, emotions, and beliefs to influence the experience of pain and helps explain why pain is a psychological phenomenon as well as a physical one. Gait control and other theorists have traditionally viewed pain as solely reflecting the action of neurons. However, the immune system also plays a role in pain. Recent research has shown that glial cells, which structurally support and service neurons within the spinal cord, are involved in the creation and maintenance of pathological pain. These glial cells become activated by immune challenges, viral or bacterial infection, and by substances released by neurons within the pathway. They then amplify pain by releasing cytokines. They are messenger molecules that promote inflammation. This may help account for that ache all over sensation that many of us experience when we are ill. The endorphins. In 1680, an English doctor wrote, "Among the remedies which it has pleased Almighty God to give man to relieve his suffering, none is so universal and so efficacious as opium. Opiates, such as opium, morphine, and heroin, have been used for centuries to relieve pain, and they strongly affect the brain's pain and pleasure system." In the 1970s, scientists discovered that opiates produce their effects by locking into specific receptor sites in brain regions that are associated with pain reception. But why would the brain have built-in receptors for opiates, unless there was some natural chemical in the brain for the receptor to receive? Later research disclosed what had to be true. The nervous system has its own built-in analgesics, painkillers, with opiate-like properties. These natural opiates were named endorphins, which is a key term meaning endogenous or internally produced morphines. Endorphins exert some of their pain-killing effects by inhibiting the release of neurotransmitters involved in the synaptic transmission of pain impulses from the spinal cord to the brain. Endorphins are of great interest to psychologists because they may help explain how psychological factors in the head can have such strong effects on pain and suffering. In 2001, John Carr Zubietta and colleagues published a landmark study that showed the endorphins in action with the brain. They injected a radioactive form of an endorphin into volunteer participants, then stimulated them with painful injections of salt water into the jaw muscles. Brain scans allowed the researchers to see which areas of the brain lit up from endorphin activity and to relate this activity to pain reports given by the participants every 15 seconds. The scans revealed a surge of endorphin activity within several brain regions, including the thalamus, the sensory switchboard, the amygdala, an emotion centre, and a sensory area of the cortex. As the endorphin surge continued over 20 minutes of pain stimulation, the participants reported decreased sensory and emotional ratings of pain. Acupuncture is an effective pain reduction technique that may ultimately be understood in terms of endorphin mechanisms. Injections of naloxone, a drug that counteracts the effects of endorphins, greatly decreases the pain-reducing effects of acupuncture. This suggests that acupuncture normally releases endorphins to blunt pain sensations. The body senses. We would be totally unable to coordinate our body movements were it not for the sense of kinesthesis. key term, which provides us with feedback about our muscles and joints positions and movements. Kinesthetic receptors are nerve endings in the muscles, tendons and joints. The information this sense gives us is the basis for making coordinated movements. Cooperating with kinesthesis is the vestibular sense, which is the sense of body orientation or equilibrium. That's a key term as well. The vestibular receptors are located in the vestibular apparatus of the inner ear. One part of the equilibrium system consists of 3 semicircular canals, which contain the receptors for head movement. Each canal lies in a different plane, left, right, backward, forward, or up, down. These canals are filled with fluid and lined with hair-like cells that function as receptors. When the head moves, the fluid in the appropriate canal shifts, stimulating the hair cells and sending messages to the brain. The semicircular canals respond only to acceleration and deceleration. When a constant speed is reached, no matter how high, the fluid and the hair cells return to their normal resting state. That's why aeroplane takeoffs and landings give us a sense of movement, whereas cruising along at 800 kilometres per hour does not. Located at the base of the semicircular canals, the vestibular sacs also contain hair cells that respond to the position of the body and tell us whether we are upright or tilted at an angle. These structures form the second part of the body sense system. You have now learned a considerable amount about the principles underlying stimulus detection and transduction. As the following Applying Psychological Science section shows, these principles have not only informational value for understanding how our sensory systems operate, but also applied value in helping people with sensory impairments. Applying Psychological Science. Sensory prosthetics, restoring lost functions. Millions of people suffer from blindness and deafness and live in sightless or soundless worlds. War, accidents or illness result in amputations that cost others important aspects of their sense of touch. Psychological research on the workings of the sensory systems is now being combined with technical advances in bioengineering, resulting in sensory prosthetic devices, which is a key term, that provide sensory input that can, to some extent, substitute for what cannot be supplied by a person's own sensory receptors. In considering these devices, we should remind ourselves that we don't see with the eyes, hear with the ears, or feel with the touch receptors. We see, hear, and feel with our brain. The nerve impulses sent from the retina, the organ of corti, or the skin are no different than those sent from anywhere else in the body. Seeing with the ears. One device, known as sonic guide, provides new eyes through ears, capitalising on principles of auditory localisation. The sonic guide works on the same principle as echolocation, the sensory tool used by bats to navigate in total darkness. A pair of eyeglasses contains a transmitter that emits high-frequency sound waves beyond the range of human hearing. These waves bounce back from objects in the environment and are transformed by the sonic guide into sounds that can be heard through earphones. Different sound qualities match specific features of external objects, and the wearer must learn to interpret the sonic messages. For example, the sound's pitch tells the person how far away an object is. A low pitch signals a nearby object and becomes higher as the distance to the object increases. The loudness of the sound tells the wearer how large the object is, and the clarity of the sound, ranging from a static-like sound to a clear tone, signals the texture of the object, from very rough to very smooth. Finally, the sound localization principle described earlier tells the person where the object is located in the environment by means of differences in the time at which sounds arrive at the two ears. In the first lab tests of the sonic guide, psychologists Aitken and Bauer used the apparatus with 6 blind babies who ranged in age from 5 to 16 months. Within hours or days, all of the babies using the sonic guide could reach for objects, walk or crawl through doorways, and follow the movements of their own hands and arms. Moreover, abilities such as reaching for objects, recognising favourite toys, and reaching out to be picked up when their mother, but not someone else, approached, seemed to occur on the same developmental timetable as in sighted children. Aitken and Bauer concluded that blind infants can extract the same information from sonic cues as sighted babies do from visual cues. Older children and adults can learn to use this device too, but not as easily as babies can. Children trained with the device can easily find objects, such as water fountains and specific toys. They can thread their way through crowded school corridors and even play hide and seek. The Sonic Guide is now being used by visually impaired children in schools and other natural settings, as well as by adults. The Seeing Tongue. Paul Bacchierita developed a tactile tongue-based electrical input sensor as a substitute for visual input. The tongue seems an unlikely substitute for the eye, hidden as it is in the darkness of the mouth. Yet in many ways it may be the second best organ for providing detailed input, for it is densely packed with tactile receptors, thus allowing the transmission of high-resolution data. Moreover, its moist surface is a good conducting medium for electricity, meaning that minimum voltage is required to stimulate the receptors. The current stimulator, shown in figure 5.23a on page 171, receives digital data from a camera and provides patterns of stimulation on the tongue through a 144 electrode array. The array can transmit shapes that correspond to the main features of the visual stimulus. Initial trials with blindfolded, sighted people and blind people show that with about 9 hours of training, users can read the letters of a Snellen eye chart with an acuity of 20/430. A modest but noteworthy beginning. With continued development, a miniature camera attached to eyeglasses will transmit wireless data to a more densely packed electrode array attached to a dental retainer. In addition to helping people who are blind, the device could have both military and civilian applications. For example, it could help soldiers locate objects in pitch-black environments, such as caves, where night vision devices are useless. It could also aid firefighters as they search smoke-filled buildings for people to rescue. Bionic eye. Vision is, arguably, our most important sense, and losing it can be particularly debilitating. Blindness typically results from damage to the retina and can either be an inherited pathology, as in retinitis pigmentosa, or age-related, as in macular degeneration. Age-related macular degeneration is responsible for about half of all legally defined blindness in Australia. The primary aim of attempts to restore sight is to develop an artificial or bionic eye. In 2009, the Australian Research Council awarded the Bionic Vision Australia Consortium $42 million to develop a bionic eye. Their approach consists of using a camera attached to a pair of glasses to image the visuals seen and then transmitting that information via radio waves to a microchip implanted in the eye. Electrodes attached to the chip convert those signals into electrical impulses, emulating the output of the retinal ganglion cells. This information is then sent to the optic nerve. The visual areas in the cortex interpret these signals as images in the normal way. While a degree of vision will occur, people using the device will not perceive the world as people with healthy eyes do. The amount of detail they will be able to see will depend, in part, upon the number of electrodes used in the device. 2 prototypes are being developed, one with 100 electrodes and another with 1,000 electrodes. The 100 electrode device should allow patients to perceive the outline of shapes and hence allow them to move around more independently. The 1,000 electrode device will provide a more detailed percept of the world, possibly even allowing patients to recognise faces and read large print. However, for comparison, there are about 6 million cones in the typical human eye. Cochlear implants. The cochlear implant is designed for people suffering from nerve deafness who cannot be helped by the mere sound amplification provided by standard hearing aids. The first cochlear implant was made in 1964. This was a 6-channel device that allowed people to recognise simple tunes. While a great deal of work was done in the next 20 years by various researchers, the major breakthrough occurred in 1978 when a 22-channel implant, developed by Australian researcher Graham Clark, allowed a previously deaf patient to hear and understand speech. The company Cochlear Ltd was formed to further develop the device and manufacture it, and today the Australian cochlear implant system is the world's most widely used system, helping more than 30,000 people in 50 plus countries. A set of 22 electrodes is implanted in coil-like fashion around the cochlea in order to directly stimulate the auditory nerve. A microphone sends sound waves to a processor implanted in the bone behind the ear, and the processor breaks the sound down into its principal frequencies and sends electrical signals to cochlear areas associated with particular frequencies. With a cochlear implant, deaf people can hear everyday sounds such as sirens, and many can understand speech. Although the substitution of 22 electrodes for the more than 16,000 hair cells that populate the intact cochlea can't produce a standard auditory experience, cochlear implants have helped many people and partially restored their sense of hearing. The bionic hand that restores tactile sensations. In 2009, researchers in Sweden and Italy announced the development of the smart hand, a prosthetic device that restores the sense of touch to people who have lost their hands. The smart hand contains 40 sensors that are connected to the sensory nerves in the arm of an amputee. 4 motors, also linked to the brain through their attachment to motor nerves in the arm, allow patients to move the fingers in very precise ways. This is the first prosthetic hand that allows the level of control of movement that comes only through tactile feedback. With it, an amputee can actually experience the feeling of stroking a loved one's cheek and can handle delicate objects with just the right amount of pressure. Sensory prosthetics illustrate the ways in which knowledge about the organisation of the visual cortex, sound localisation, and the place theory of pitch perception can produce the information needed to take advantage of new technological advances. Yet even with all our ingenuity, prosthetic devices are not substitutes for our normal sensory systems, a fact that should increase our appreciation of what nature has given us. Concept check. Skin and body sensors include touch, kinesthesis, and equilibrium. Receptors in the skin and body tissues are sensitive to pressure, pain, warmth, and cold. Kinesthesis is mediated by nerve endings in the muscles, tendons, and joints. The sense organs for equilibrium are in the vestibular apparatus of the inner ear. Pain receptors are free nerve endings, and the sensation of pain is influenced by downward influences from the brain, gate control theory. Endorphins decrease pain. Knowledge of the sensory mechanisms has been used in the development of sensory prosthetics for people who are blind or hearing impaired or who have lost one or more limbs.